Are on.
All right. Well, thank you all for attending today on a Friday. I regret that I'm not there in person. I really wanted to be there, but as you know, there's some bugs flying around. I don't know which one it is, but I have a sore throat and I didn't want to risk it. So thank you all for coming and attending this virtual session from my side. So today I'll be talking about omics data exploration, and really when I was first asked to present about multi-omics data analytics, I couldn't really talk about multi-omics without breaking it down into a bunch of different topics. And so the next couple of workshops will really be around omics data analysis and looking at it in different ways. And today is going to be just about exploratory data analysis, not about like multi-omics data integration or anything. So I'm a new faculty in the Department of Anesthesiology, Pharmacology, and Therapeutics, and mostly a computational biologist. And in the next an hour and I guess like 15 minutes now, I'll sort of walk you through various ways of how you can explore your omics data when you get it. But first, we want to begin by acknowledging the land that we gather. We want to acknowledge the traditional, ancestral, and unceded territories of the Musqueam people. I also live on near campus, actually, Westbrook Village. So this is represents where I live accurately. Copyright information. This is copyrighted by CC BY-SA 4.0. And just for those of you who don't know what is required about that, I think you just attribute the users, the creators, sorry. So very targeted learning outcomes for today. And there's sort of fall more around more of the appreciation, first of all, about why it's important to do exploratory data analysis, and why it is a prerequisite for any research study that you do, instead of going straight into your question of interest. Oftentimes, when you don't look at the data carefully, you sort of miss various aspects that might be affecting your data, like batch effects, outliers, and things like that. And so it was a good idea to look at those type of questions initially. The workshop today, I purposely incorporated Python and R code just to give an extra flavor of what types of analysis that can be done in these two ecosystems. And personally, I don't favor one or the other. I use both. And I recommend that you also try out both languages for your analyses. And there'll be a heavy focus today on dimension reduction techniques. And essentially, it's a way of taking a look at your high dimensional data and being able to visualize these high dimensional artifacts in lower dimensional spaces and in like 2D scatter plots. Phil, is my voice coming through okay?
Oh yeah, we hear you fine.
Okay, perfect. So as I mentioned, today the workshop is sort of using a different approach than the previous workshops, which were mainly focused, I believe, mostly in R. And so the image that was being used initially did not have Python installed. So depending on how you were attending this workshop, I have instructions for both sort of going through this workshop either on your local machine or on Sockeye. For your local machine, you would need Docker installed because the image that we're creating is custom. And you can find this image if you clone today's repo. This is Phil's repo. It's called Precision Health Virtual Environment. Once you clone it, you can go into this specific workshop folder. Then you can create the repo, and you can also host the repo on your local. If you remember when I gave the GitHub lecture, there's a GitHub username, but now for Docker, there's a Docker Hub. And once you have that hosted, then you can use it on Sockeye. right? But for the purposes of this workshop, if you have already logged into Sockeye and you have made your own student space from the previous workshops, then all you have to do is copy the today's workshop folder. And if you've attended past workshops and you're sort of familiar with this already, so you can go ahead and do that as well. I think when I had just done it right now, I believe it's one of these terminal windows. Let's just see the path. This is projects, not the window. So I have basically done those steps on my side. So here I copied the workshop from the project directory. So if you can also copy this command, then you would get all of the material as well. And then what you need to do is there's a file there that's called run rpy studio, and you need to modify this and input your email address, and that's pretty much it. And you should be able to run the job script, and it will give you the connection file that has the SSH link in it. You can ignore the rpystudio.sh file and just focus on the run rpystudio.sh. Any questions on that?
Maybe just pause to let people set it up.
How do I install Docker on RStudio on my local computer? If you are in my if you go to my slides, you'd have to download these slides. They're on GitHub, but you need to have GitHub installed first, right? So if you're able to install GitHub Docker, sorry, I don't know why I said. I think I have the instructions also written on the README page. So if you go to the this link, I can actually post it in here. Then the first thing to do is make a Docker Hub account. And you also have to download Docker and install it, because you need to be able to work with Docker to be able to create the image. And I think you can just type in like Docker install or something. But that's sort of beyond the scope of today's workshop, because it's already there on Sockeye. So if you're on Sockeye, it should run for you already. And once you have the job running, it opens up an RStudio environment. And if you remember from previous lectures, there is an important step about setting the library paths, so it knows where the installed libraries are. So you'd have to do this for your own user area. And I can put this in the chat as well. And so just modify the CWL with your CWL, and it should be all set to go and access the packages that are already installed there. And then what I did was I clicked knit, right? And since I've tested this already, it should create the entire talk for you for today. And it looks something like this. Any questions? And a lot of these steps, I know I went through it fast, but it's pretty much what we've been doing for the last 2 weeks, right? So hopefully it's not too much of a hassle of getting the RStudio session running. Can anyone give me a feedback on whether they were able to run it?
You want us to knit it too?
Yes. Once you've set the library press, please press the knit button. And I think there should be no hiccups. And so the reason why I've created it the way it is because I don't really want people to be writing code in the session, because it's all being run all at once, right? So the fact that it's once you press knit and it all run and it runs perfectly means everything works. And so then it just I focus more on the material and sort of show you the code and walk through instead. So Phil, if you're able to run it, let me know if there's any hiccups you get.
I had an error in facto extra.
Did you set the lib paths?
Oh, no. Good point. There it goes.
And so that's one way of creating the slides. Another way of creating the slides is this is weird. That's weird. That's interesting. So let me know when that finishes, and then I can keep going, because then I'll know that it's working for at least you. Oh, no, it should take some time at a part of the workbook, because it's doing a method that takes some time. It's called t-SNE. But other than that, it should run fine.
So the output of this is going to be just the PDF that we already have.
Yeah, but it's gonna open up in your like another window.
Okay.
But you can open it up in your browser as well. But the PDF is basically like printing out the browser output.
Okay. So people could just follow along with the slides if they can't get the knit to work.
Exactly, yeah. But as you can see, I keep rerunning it, and it is working, right? So
yeah.
Hopefully it's working for you as well. But in the interest of time, I'll keep going. So if you don't have Sockeye, and if you don't have it running, then the rest of the talk should also flow. All right, so I'll keep going then. So as you all know, there's no surprise here. Omics data is quite ubiquitous in biomedical research, and so there's a lot of disease domains looking at omics data, whether it's gene expression or other types of data sets. How do I hide this again? Disable. And so these are just a few examples of where they've looked at, especially like genetics and gene expression signatures. And all of these analyses were like retracted or highly criticized because of things called batch effects, right? So technical artifacts that had affected the data and sort of confounded the results. And so a lot of the things that people were finding were actually due to technical artifacts, not because of the biological variable of interest. So here's an example of that. This was a study where people essentially did, I guess, gene expression on different organs of both mouse and humans. And you can see them labeled in this table here. And you can also see that the red, which is the human samples, were run in separate lanes of the workflow compared to the mouse. And so when you did sort of like an unbiased clustering, then all of the organ samples from each species clustered separately. And so this was sort of a result where they were arguing whether or not the mouse is a useful model system of the human, right? So, but as you can see, it was sort of confounded by the batch here, right? So once you sort of take account of the batch, then you have the individual organs from the both species clustering together, which you would actually expect, right? Because a heart from a human is more similar to a heart than a mouse than it is to other organs in the same species. I'm sure people are probably familiar with this error that has come up across many times that you might have experienced, right? So gene names, and Excel is notorious for modifying things that you open up in Excel. And so one of the biggest problems that has been observed is essentially just the names of genes being modified. So here are just a few examples of things that can go awry. So for the purpose of this talk, I'm not going to be going into like QC and data pre-processing of omics data, just because depending on the omics modality that you have, the steps leading up to what the normalized standardized data looks like is very different, right? So if it's like a protein mass spec pipeline, it's very different than a gene expression pipeline, and it can depend on the type of technology used. So for the purpose of this talk, I'm just going to assume you have matrices of numbers, and then what can we do with that? And so this matrix of numbers, as I mentioned, can be represented by a letter here. So this is represented in this green rectangle called X. And different communities like to arrange this matrix in different ways. And so on the left here, we have your X matrix that is of dimension number of samples on the rows, and then the columns are the number of variables, so like number of genes, proteins, etc. For this other community, which is the R community, the samples are on the columns and the variables are on the rows. So as you can see that they're transposed. And so these two blueprints you can think of for representing these omics data. One is for a package called ANData, which is in Python, and the other one is called Summarize Experiment, which is in R. And so different, and this is just because of historical reasons, why they're represented in slightly different ways. And what's really interesting about these bioinformatics tools is that not only can you host these data matrices, but you can actually host a lot of other data attributes along with it. So for a given X matrix, you have a lot of information regarding the individual variables, individual observations. How are those variables related to each other? So even like a pairwise correlation matrix, you can hold that for both the observations and the variables, right? And so similarly in the R space, you have a lot of metadata associated with your samples. And so you can see how easy it is to work with some of these standardized tools where you're not just working with the data matrix, you're actually working with a lot of other, I guess, attributes of the data. And this becomes really useful when you start applying various functions to it, because then different functions require different attributes of the data, and they can just access through all of that through one interface. And you might have sort of seen this in single cell analysis in like Seurat, for example. I always like to review terminology just because there's a lot of redundancies in what people refer to various terms here. So variable can also be described as a feature. You can also think of a variable as in like an axis, or in other words, a dimension. And so a common types of variables include age, sex, various genes, proteins, SNPs, OTUs, pixels, right? So there's a lot of types of variables that exist. On the other hand, observations are what you actually collect these variables on. And so observations include individual patients, subjects, cells, mice, so on. And for the purpose of this lecture, since we are talking about dimension reduction, where you necessarily are interested in computing variables that are not directly observed, but are actually computed from mathematical models. And so they also have a lot of different names. So they can be called components, factors, like hidden variables, latent variables. So all of these things are pretty much the same thing. I don't see any questions. Yes. Phil, just fill me in if there's a question.
Yep.
All right, so we're gonna start with a use case, and this is just data that I've generated just using some code. And here, for the purpose of the presentation, I don't show you the code for it, but you can see the code in the vignette. And so all of these slides contain the contents of the vignette. So I think it's somewhere up here. Sorry, it's way up here. So this is the terminology, and just a quick recap about the way that this is structured. These three dashes just encapsulate one single page of the slide. So the page that I'm on right now, which has the fake data, is actually this part here. And I've actually set echo to false, which means it's not going to display the code. And so this, you can look at the code here. It's not really relevant for the purposes of this talk, but we have about 100 observations and 1,000 variables. And for the purpose of this simulations, I have 5 like outliers that I have added to the data set, right? So this is what the data set looks like, and then now it's like, what can you do with this data to find different patterns that may exist in this data set? So the first thing you could do is look at classical techniques like making like histograms, box plots of your different observations or variables. And so right here you can see that just by making box plots of the data for each individual sample, I'm able to get like an idea of the spread. And you can see that some of the observations already, some of the samples have a much wider spread, right? So already we're able to identify these different observations. You can also like randomly pick genes and look at what the distributions look like. And so a lot of this stuff becomes very challenging when you're looking at the high dimensional space. This type of work is very useful when you have only like 1 or 2 variables, but as you can see, it's very difficult to, even with this classic approaches, to find any meaningful patterns. A useful technique is the sample correlation matrix. So you can just take your data matrix X and just multiply by itself. And if it's centered and scaled, you get something called a correlation. And so here you can look at what the individual correlations look like. So the correlation ranges between -1 and 1. And so if you have, since you are looking at all of your variables when you're computing these correlations, then broadly speaking across all variables, you shouldn't see any like major deviations from the correlations. But you can see here that there are some, if you see some of these lines that sort of pop out at you, you can see that they're very different from the rest of the data that exists there, right? So these are those outliers I had incorporated in my data set. And oftentimes it's a lot more striking. Like you'll get this clustering that you see, this tree clustering, where it would pick out like a few observations that cluster very differently. And those are clearly outliers, maybe like something went wrong with the machine or something. But it would be really nice to get a plot where you can just get all of the observation laid out. And you can clearly pick out some of these outlying terms, or even like color code this and see, is there any like clustering pattern that exists, right? So this is sort of the a lot of the work in exploratory data analysis has gone into. And so this is what brings me to dimension reduction methods. And essentially it's a way of visualizing your high dimensional data. So if you remember in the previous example, I had like 1,000 variables, right? And so I'm visualizing all of those observations instead of in 1,000 dimensional data cloud, which is like you can't visualize that in a 2D plane, right? So how can we condense all of that information? Or in other words, how can we map this high dimensional data cloud into a lower dimensional space and this like 2D space, and still retain a lot of the structure that's associated with high dimensional space. So like similar observations that are close to each other should be similar in this high dimensional space as well.
There's a
and
question
yeah.
in the chat. Does clustering mean correlation?
No, it does not. So if you look at this plot here, when I had computed the correlation, right? The correlation just tells you how associated two, I guess, vectors or two things are related to each other, right? So these correlations could be samples or they could be variables, right? So if a correlation is of 1, that means both of those variables or samples explain the same thing, or maybe they are the same thing. You can then use something called the correlation matrix. So what I have done here is I've taken all of the 100 samples that I had and computed all combinations of pairwise correlations, right? And you get a matrix that looks something like this. But then you can use that correlation matrix to start seeing which samples have similar correlations with each other, and then that's where you would get the clustering aspect. So correlation is not necessarily clustering, but things that are correlated will cluster together once you do cluster it. That makes sense?
Yep.
Okay. All right, so just looking at the the dimension reduction approaches for high dimensional data, and I found this really nice review on tips on effective dimension reduction. I highly recommend reading it, just because it sort of describes some of the historical aspects of where these methods were coming from. And so you can see there's a lot of methods here depending on whether your input data is of various types, right? So the classic, very conventional and I would say like very robust method is PCA, continuous data. But then you have other methods that exist for other types of data sets, right? So if you have categorical data, you can't necessarily apply PCA to it. So there's variations called correspondence analysis, and I'll sort of speak a bit about that later on. We also have some of the newer age approaches like t-SNE and like I think some of the deep learning methodologies, which are very, I don't think, oh yeah, so it checks off here. So these are a lot of like non-linear methods. So these can sort of identify sort of these clustering patterns that is not necessarily captured by some of these other linear-based methods, and we'll see examples of these as well. But all of these are something called unsupervised, which means that they take your data as it comes, and they just try to represent your data as a whole in a lower dimensional space. These other methods at the bottom are supervised in that there's this other variable called the Y variable, and that is used to inform how the points or the observations are arranged in this lower dimensional space. And so that's why it's called supervised, because you're sort of you're taking a peek into some of the labels of the individual observations. And there's two methods here. These are multi-domain data when you have multi-omics data, which we'll get into on Monday. So this effort was led by Susan Holmes from Stanford, a well-known statistician in the field. And so the first tip sheet that they recommend in the paper is choosing an appropriate dimension reduction method. And so this can be very important in representing your data in this lower dimensional space. And this is really dependent on what your data looks like, right? So if your data, and if you suspect that your data is sort of this like cloud of points, which is sort of following like an ellipsoid pattern, then PCA is ideal for that type of data set. TSNE is more of a non-linear dimension reduction method, and it's very interested in grouping points that are in the neighborhood of each other. And so one of the parameters that it takes is something called perplexity, which is essentially like an average number of neighbors that it has. So within these TSNE clusters, the individual points are very related to each other, but there's no concept of meaning to the distance between clusters. So it's actually meaningless to interpret a distance between clusters than it is within clusters. A more recent approach called FATE, which is based on distant matrices, and there's like a bunch of transformations involved. I recommend reading it. It's very useful for like differentiation patterns or like these tree-like structures that exist, right? So you can see here that for a simulated artificial tree, PCA does a pretty good job of describing what this tree looks like, right? But you can see that it's trying to still decode this tree into this like ellipse looking thing where you have these like major and minor axes. TSNE sort of loses some of that tree shaped structure because it's more interested in computing like neighborhoods of similarity, not trying to explain like the global structure, whereas FATE is trying to do both, right? So depending on the data set that you have and what you expect from the data set, then you focus on a different approach. In my experience, I try to use a bunch of these methodologies. Obviously, there's some that are not recommended. PCA is not recommended for single cell data, and there's some variants that have been applied to make it applied to single cell data. And so I think if you're familiar with Surat, that was covered, there's a SC transform that is used to modify the data prior to PCA. And so since we're sort of talking about PCA, let's sort of look at some of these dimension reduction approaches in more detail. So the first one is PCA, and the way that you can think about PCA is a reformulation of your initial data set. And so here I've written PCA, which is I've written PC. Think of this as a matrix of data. And this equals your original data matrix, where your matrix X, and it's essentially being weighted by different values, right? So all of your individual variables get weighted differently, and that is what creates a principal component, or a PC. And essentially what it's doing is that if you have a higher dimensional space, which is represented in this cube here, we can find these axes of that sort of capture the majority of the variation that's in your data set, right? So the axes will sort of align with wherever that variability exists. And so you can see here that we're going from a 3-dimensional space to a 2-dimensional space, but we're still capturing the global structure of this cloud of points in this lower dimensional space. The reason why this is very useful is because your principal components are actually just your original data set, but they've just been weighted differently depending on the individual contributions of these original variables to your principal components. And so another way of looking at principal components, you can think of them as these unobserved variables that exist, but they're not directly measured, right? So, for example, pathway activity, for example, right? You have all these biological processes happening. You can't estimate them directly by measuring, like quantifying the level of a pathway, but you can measure by individual members of that pathway, like genes or proteins or so on. And then you can think of these PCs as combinations of those individual variables. And so the weights are dependent on which individual genes or proteins belong to those pathways. And that's sort of one way you can look at interpreting some of these principal components. So this is another description of principal components and how it's really trying to represent your data in a different way. And so this figure A is just showing you an example of individual genes. We have like 3 groups of gene patterns, right? So A, B, and C have a pattern that looks like this. D, E, and F have a pattern that look like this. And then G, H, I have a pattern that look like that, right? And essentially what PCA is trying to do is it's trying to find sort of these global trajectories that can summarize multiple patterns using a single variable, right? And so this becomes very useful when you look at diagram D here, where as you have more and more principal components, you can recreate your initial data set. And so this is PC1 here, but as you add more and more PCs, you can see that you're able to sort of reconstruct your original data set here, right? So you can see here that this profiles look exactly the same as this one. And the reason why this is very useful is because initially we had 3, 6, 9 genes, right? But now we have 3 principal components that are able to capture the gene expression patterns in those 9 genes. So we have, in essence, reduced the dimensionality of our data set because there's a lot of redundancy in high dimensional data. And so instead of working with a data set that has 9 genes, we can now work with a data set that has 3 genes, right? And you can actually see the proportion of variation explained by those individual PCs. And this plot is actually telling you that within the first 2 principal components, you're able to explain the majority of the variability or the information that's contained. And that becomes very useful because now you can actually just plot your observations using those 2 PCs. And you can see, hey, these groups of genes exist without actually looking at like thousands of these patterns, right? You have a simple 2D visualization that depicts similarity between observations. Any questions on this? Okay. So if you ever have used PCA, under the hood, it's really trying to like refactor the data, essentially using a bunch of products of matrices, and they have very special meanings. And I recommend you looking into something called singular value decomposition, a very popular factorization method to sort of decompose a given matrix to a product of three matrices, where you have your first two matrices, which represent your PCs, and then you have your matrices that holds essentially weights for the individual variables. And so this is just if you're familiar with R, PR comp is a like very, I don't want to say old, but it's a very established function to compute principal components. And you can look under the hood of PR comp by typing in methods PR comp, and then it'll give you some of the methods associated with this class. And you can get the individual formula for how this is actually being computed. And if you look under the hood, there's an SVD call that is computing those principal components. And so the reason why this is very useful is because there's a lot of nice properties with PCA, like the diagonal matrix, for example, contains various variances associated with the PCs, and they sort of describe the information that's being captured. Another useful or may or not be is that the pieces are uncorrelated, which means that the first principal component has zero correlation with the next one, right? So the individual PCs are independent and they contain independent information from one another. How is principal component different from principal coordinate? I'm not sure. There's a lot of variations, like co-inertia analysis and things like that. And there's also factor analysis. I feel like there's a lot of similarities between them, but I can't tell you in terms of like the math similarities or differences. I'm not familiar with principal coordinate. Okay, so we're gonna try to use a single data set to apply some of these methodologies, and there's one that's already in one of the R packages. It's called Iris. And so I believe it's in the, if you want to know more about this data set, you can type in question mark and then Iris. And I'll give you a description of what that data is. Essentially, it's a data set on 50 flowers across 3 species. And it has a bunch of variables like the length of the sepal, petal length and width, and so on. And the reason why I chose it is because it's already built in, and it's very tiny. So you'll see how it's being how PCA is sort of capturing this this data set that has 4 variables in like a lower dimensional space. So these are the individual variables, right? Sepal length, width, petal width, length. And I've just done a correlation plot. You can see the code for it in the R Markdown. And I'm just color coding them based on the different species. So this clustering is it's not driven by this the species variable, right? So it's like a ad hoc thing that I've used to cluster. I've used to color them. And we can apply PCA to it just by running the prcomp command. And you can choose how many PCs you want to contain. But by default, I think if you don't do that, it'll compute the PC's principal component, all of them. And essentially by all of them, I mean the minimum of however many genes or however many observations or variables you have. And so in this case, we had 4 variables. So you can see that there's 4 PCs that were computed. And in this case, we're computing the proportion of variation explained. And you can see here that the first 2 PCs contain 95% of the variation, right? So all of the patterns that you essentially exist in the 4-dimensional data set, we can capture in 2 dimensions. And that sort of just speaks to how useful this approach is in sort of reducing that dimensionality, but still capturing most of the information. There is like a loss of 5%, but it's a trade-off between how much you're willing to lose versus how reduce in terms of lower dimensionality. So once you have your principal components, actually, I'll pause there. Does anyone have any questions so far after about PCA? Okay, thank you, Kang. All right, so PCA has a bunch of nice visualizations based on the outputs from that factorization I was talking about, and the Facto Extra has very nice functions that take in the output of PRComp and make some nice plots with it. And so the first one is called the component plot, and this essentially will plot the individual observations on a 2D scatter plot, and you can sort of select which PCs you want to plot. And you can see here that this is that 95% of variation being explained, and this is just all of the clustering of the points. And so this is something that exists in 4 dimensions, right? Those 4D that we talked about. There's like a 4-dimensional cloud, and we're representing that in a 2D space and still maintaining the global structure of the cloud. So which means that points that are close to each other are actually close to each other in that higher dimensional cloud. And so there's a lot of inference that you can make here, right? So already you can see that the Setosa species has a similar clustering pattern, this high dimensional cloud, than the other 2 species here in green and blue. You can also look at something called a correlation circle. And this just comes from this neat like standardization that's applied in PCA, and sort of didn't mention it initially because I'll go back to it later on. And essentially it's the art of centering your data, which means removing the mean from all your variables, and also standardizing. So scaling your data so that the standard deviation is 1. And you get this nice plot where you can actually interpret relationships between variables depending on the angle between these arrows, right? So if your angle are acute angles, then those variables are positively correlated, and if they're if they're obtuse, then they're negatively correlated, and if it's like 180 would be like direct negative correlation. So you can see here that these 4 variables are positively correlated, whereas this one is negatively correlated with the other 3. And you can read more about interpreting these plots in this paper here. Okay. Similarly, since you're able to represent your variables are in like these new coordinate systems, you can actually plot both your observations and your variables on the same plot, right? So this is called a biplot. And this becomes very useful because now you can actually take a look at those axes, right? So each individual variable is an axis, right? You can think of it as like an x-axis, y-axis, z-axis, right? And the direction of the arrow points in the direction of increasing values of that variable, right? So here, if we take a look at observation 132 and observation 42, then you can see that if you look at this direction here along sepal length, then there's a big difference between sepal length between flower 42 and flower 132, right? And so this is, don't take my word for it. So if we like sort of extract the sepal length for flower 42 and 132, which I think I did it on the next slide, then you can see here that the sepal length for flower 132 is much larger than the flower length for 42, and that we had seen this in the direction of the axis here, right? So you can see here that we're able to get a lot of information out of this high dimensional cloud just by a 2D scatter plot. And this sort of confirms that that actually is true. Any questions so far? Perfect.
Yeah, question.
Okay.
It's 90 degrees. Does that mean that there's no correlation? Like, for example, if you can go back to the angular between like sepal length and sepal width, it's about 90 degrees.
Yeah, you're right. Yep, no correlation.
So
Isn't that? Yeah, sorry, go ahead.
sorry, yeah, between those two things, there won't be any correlation then, right? Is that what it means?
Oh, between sepal length and sepal width, you mean? Yeah, that's exactly correct. Yeah. Yeah, so it's the direction doesn't really matter. So if this was on that side, it would still be 90 degrees, so it'd still be zero correlation. It's more than 90 here, so there's slight like negative correlation between these two and this one. Yeah. But so you can see there's but between these two, there'd be like very strong positive correlation between these two. So this is petal length and width, right? So if we actually go back, petal length and petal width, right? You can actually see that being validated in the scatter plot. You see that? It's like highly correlated. And that's what PCA actually picked up for you. And then going back to your observation about sepal length and sepal width, they're uncorrelated. If we go back to that scatter plot, sepal length and sepal width. So that's what the cloud looks like. And you can see that it's basically like a cloud centered, right? So if there was no like coloring here, you could technically say there's no correlation between those variables. However, once you segregate it by the different species, then you do see correlations, but PCA is unaware of that, right? So it makes perfect sense that it thinks that there's no correlation here, but there's a high correlation between these variables. Good question. All right, so I mentioned that PCA is very good at identifying these patterns, and because it's trying to like explain the global structure of this cloud of points that exists, right? But oftentimes we have a lot of like non-linear patterns that exist in the data, right? So you get like these very like spiral patterns and things like that. And this is very useful for things like handwritten digits, for example, right? Those patterns are very non-linear. And so these are just some examples where PCA would fail, right? So if you had a pattern that was like a spiral like this, and PCA is trying to fit like ellipsoid axes, then it does not do a good job, right? Or if there's like non-orthogonal patterns, like variables that are, there's like a lot degree of high correlation of the underlying variables that you're looking for. Or if you have clusters that have these, again, this is more of a non-linear pattern that exists, right? It can't sort of separate these or directly model these different neighborhoods of points, for example. And so that's where some of the non-linear dimension reduction patterns techniques will come in handy. And that was something that was also observed in single cell omics data, where PCA was shown to have poor like ability to capture the variability in the data, and the following slides will explain why that is. But before that, let's look at TSNE, which came out of Jeffrey Hinton's group, who is revered as like the godfather of AI. This is like, I think 2007 or maybe before that time. And the idea really was to focus on local neighborhoods of points. And so this technique is very interested in identifying clusters of groups, but it's very dependent on this parameter called perplexity, right? So if you sort of specify this parameter, over a various range of values, then you're going to get very different clustering patterns. And that's just the nature of the beast, is trying to optimize neighborhoods within that parameter considerations, right? So they do recommend some values between like 5 and 50, but I recommend reading some of these resources that sort of explain more of what it's trying to do, and a lot about what it is not trying to do, right? So a lot of the like cluster sizes and distance between clusters, they cannot be interpretive and they have no meaning, whereas the most of the meaning just comes from the individual groups that are identified with t-SNE. And I won't go into the math behind it, but essentially a lot of this approach comes from trying to model like complex functions with simpler functions that are easy to model, right? So you have like this p function, for example, that's very complex, so you can't really represent it. And then you essentially just find a lower bound to this function that's more easily differentiable and easy to work with. And then you essentially just use that as an approximation, and you try to minimize the distance between them. And that's where this like pretty like popular way of minimizing loss between distributions. This is like Kullback, Libero, KL loss, it's called. That's very popular in deep learning algorithms, you'll find. And this is the approach used in t-SNE. So if we apply t-SNE now to the iris data set, and this is just a function called t-SNE, and you can apply your perplexity parameters, sorry, and specify a number of low dimensions, right? So in this case, I'm keeping 2 dimensions. And so you can see how the clustering pattern changes as I modify the perplexity, right? So with a 5, you get basically like this, right? It's like very uninformative. And so you have to sort of optimize this depending on the loss, right? So you can technically plot the perplexity parameter against the loss and see where does the loss converge, and then use that parameter for your visualization. So very risky to use like default parameters for like all your data sets. All right, so another approach that you might have seen is applying both of them together, right? Apply PC and t-SNE, right? So this is a paper now a couple years old where they essentially applied PCA to single cell data. And what they found is that the first dimension was very correlated with essentially the number of zeros in your data set. And so you can see here essentially the library size, right? Which is similar. And so you can see here that if you look at the zero fraction in your individual cells, then the first PC is very correlated with that, right? And this is, and as I mentioned, a common practice is to apply t-SNE to your PCA transform data. You can see a similar effect in the t-SNE representation here, right? So there's this clustering that's driven just by the amount of zeros you have in your cells. I have a question. Remember to use set seed before everyone. Okay. Yeah, so this is actually a good point here Daniel raises to set seed. And the reason why you need to set your seed is because if you look at this distribution here, they actually start off with random values. And so these are randomly generated and they're using a T distribution. And so if you don't set your seed, that initial random values are going to be very different, right? So they might converge differently. And so setting the seed will help you get the same results every time. Sorry, so going back to the problem with PCA on single cell data. And so this is just coming from the zero inflation values and how they're affecting the first dimensions that you see. And so they, a couple of papers have tried to model the count data directly. So instead of the conventional like log transformed count data that we had started seeing with RNA-seq, they try to model the count data with other distributions than the Gaussian. So they use like negative binomial, Poisson, and so on. And so there's been a bunch of approaches looking at this. And so if you're familiar with Surratt, there's a SC transform. And so this is a review by the authors of Surratt where they sort of talk about their approach and using things like generalized linear models, which are useful for data that's not like normally distributed, that have other types of distributions. And so they treat the sequencing depth as a covariate and sort of subtract it out of the original data set. And so you can see here, once you do make that correction, then that correlation goes away, right? So that clustering is no longer affected by the zero inflation. And then it's appropriate then to use PCA, like this modified version, on single cell data. And so the common approach is to apply this like SC transform, apply PCA, and then apply t-SNE. Because SC transform gets rid of this artifact of zero inflation. PCA reduces a lot of the noise in your data set. And then t-SNE is useful for visualizing those nonlinear patterns that may exist. That's often seen in single cell data. Any questions so far?
Sorry, I'm right now here. I'm a little bit puzzled about the t-SNE on PCA or t-SNE on GLM PCA. So this t-SNE on PCA means that first of all, the raw data was the raw data utilized as the input of PCA to reduce the dimension, and then this kind of two-dimension thing, the input of t-SNE again. Does it mean that, or I'm not sure what do they mean? What do this author mean by t-SNE on PCA or t-SNE on GLM PCA?
Sure. So if you remember when they had your data matrix X, right? If you apply PCA.
Go ahead. Sorry to interrupt.
It's okay. I was just trying to understand what you're saying, but essentially what it's saying is you take your data matrix X and you apply GLM PCA, and you get a new data set that's sort of corrected for the zero fraction. And then on this new data set, you apply t-SNE to it. And the reason why you do that is because this GLM PCA reduces a lot of noise in your original data set, and it also removes this zero fraction effect that is seen in single cell data, right? So, but it's still like linear. It's still linear transformation you apply to your data set, right? And you don't get any like nonlinear clustering patterns with PCA, right? So when I talked about these types of things, PCA cannot uncover that. It's very good at reducing noise though, right? So it's still very useful to apply this, because if you remember from single cell data, these matrices are like, they have like 100,000 variables that can be profiled at the same time. And with PCA, you can shrink that down to like 50 or 100 or something, right? And so you're reducing a lot of noise that may exist, and you're also making it a lot faster as well. And so it's a common approach to apply PCA, or this like modified version of PCA called GLM PCA, on your count data for single cell data, and then apply t-SNE to it.
Okay, so here the output of PCA or GLM PCA are the input of t-SNE, and then draw this kind of two-dimensional figures on the bottom box, right?
Yeah.
That's the second.
Yeah, so that's basically what this paper proposed. In Surratt, they have another correction function. It's called SC transform. So there's like another way of doing it, but it's basically both using the same like underlying like methodology around generalized linear models.
Okay, this SC transform is used the GLM PCA, right?
No.
They are
SC transform, I think the difference is that they say our SC transform uses Pearson residuals from negative binomial, and GLM PCA is focused on like Poisson distributed errors. They're very similar. One is just accounting for like over dispersion. But in practice, there's still like a lot of like hyperparameter tuning and things like that. So I would apply like both of them to it, right? Any other questions? Okay. And so now I want to throw another dimension reduction method, and there's like so many out there. I don't actually know all of them and know all the math, all of them. Otherwise that's all I would do. But there's a nice paper and this methodology on sort of building this manifold, trying to construct a 2D representation using distance matrices. And I think I mentioned the reference. Sorry, I guess I don't have it. Maybe it got cut off at the bottom, but it's probably in the R Markdown, the paper for it. And it's called Fate. And a lot of the simulation studies that I saw in the paper were around like tree-based looking patterns, right? So if you're ever looked at like differentiation patterns for like single cell data, then this method is I would say very useful for identifying those kinds of patterns in your data set. And so this is initially written in Python, but now it has an R package as well. So this R Markdown should run the Python code as well. So one of the reasons why I created this custom image is so you can run both R and Python code as well. And so this is the output of what that dimension reduction looks like. And you can see it does have this like tree-like branching property. And it claims to model like the global and local structure at the same time. But you can see there's clearly things that are useful in each of the individual approaches that we're using here, right? So there is some sort of spread that's informative, I would say, of the individual approaches. For example, for PCA, like this observation is very different from this observation, and I think what Fate has done is like really compress the data into these like tree-like structures, right? So it's very hard to sort of talk about spread in these representations. So it's almost like these dimensional reduction methodologies, since they're sort of looking for different things in your data, it's often a useful approach to apply multiple of these things and see what comes out of it. All right, so this is another table around what type of input data these approaches like, right? So a lot of data sets like to use just the raw counts. And so these are just the individual methods on the left here. Some methods do like getting pre-processed by PCA, right? So I mentioned PCA and looking at the top number of PCs. TSNE can also look at PCA process data, and so as UMAP, right? So if you're familiar with Seurat, they do SE transform and apply PCA and then apply UMAP, right? So you can apply other combinations of transformations here as well. Thank you, Kang. And so the second tip actually for that paper was knowing how to pre-process input data, right? So depending on your approach, your pre-processing can be very different. So for PCA, it assumes that your data for your matrices are centered, which means you subtract the mean from the individual variables. And essentially the effect that it has is that it shifts the origin or the center of the data cloud to the origin. And that's important in actually the math behind it. Also, if you scale your variables to have unit variance, then what you actually end up with is a correlation matrix that you're actually applying PCA to. And then a lot of those correlation circles and those things become very useful as well. T-SNE and FATE can work with counts, but as I mentioned, you can also pre-process them with PCA to reduce the noise in your data and also to speed up the computation of your data, of the method, sorry. So a lot of that methodology was done on continuous data, but there's also approaches useful for categorical data. And so the approach around categorical data is really to encode your categories with zeros and ones. And so if you're ever use lemma or are familiar with, I think, Hessian matrices, where you can, there's a typo there. You can encode a single categorical variable with zeros and ones. And so what you actually end up with is if it's a binary variable, one category is going to be all zeros and the other one's going to be all ones. If it's a categorical variable with multiple, like more than 2 categories, then it's going to actually end up with multiple columns in your matrix, because you do want zeros and ones. And this actually inflates your dimensionality of your data set, because now you got a bigger data set than original. And there's this same sort of factorization, still singular value decomposition, but there's some corrections that are applied for those variance measures and so on. So that's called a multiple correspondence analysis. It can be accessed through this package here. And following that, there's also a multiple factor analysis approach where you have mixed data types, right? So you have often seen with like clinical data where you have continuous and categorical. And the approach there is to apply PCA and MCA separately to those individual data matrices and then combine the resulting components. So highly recommend that you check out these packages that apply these approaches. And a lot of these approaches are very been used for a long time, right? So they're very established methods, actually. So tip number 4 is to use the appropriate measures to compute similarity and distance. So depending on, again, your data, you want to use a different method to compute your distances. And this is just some recommendations. You can read more about it in the paper. So if your data is not continuous, is binary, and there's something called a Manhattan distance. If it's sparse, a lot of categories, then use something called Jacquard. If you have mixed data types, there's something called a Gower distance, right? So depending on the type of data set you have, this really doesn't apply to like single cell data, but if you have like a lot of clinical data that has a lot of different types of variables on different scales and different types, and this type of stuff becomes very useful. Also, for dimension reduction approaches, you want to decide on a number of dimensions to keep. For PCA, it's actually very straightforward because you can plot the proportion of variability explained for the individual PCs based on something called the eigenvalues. And so here you have this plot that looks like this, and you can see that the larger the bar, the more variability it explains. So there's this thing called the elbow point where you can sort of use that as a visual inspection and keep those PCs that have like a substantial amount of variation explained. For other metrics, methods such as T-SNE, you can choose the dimensions depending on the KL loss, as we talked about earlier, and then choose your number of dimensions based on that. Yeah. Okay, cool. So the 6th tip that they make here is applying correct aspect ratios to plot. So I mentioned PCA. This, I would say, applies mostly to like PCA, where the individual dimensions are explaining a different level of information. for your data, right? So the first dimension was 73%, and the second dimension was 22%. And so you want to make this proportional. So instead of having square plots, you have a plot where the axes are sort of scaled appropriately. So in this case, the x-axis is longer than the y-axis. For other techniques like t-SNE and UMAP, that doesn't necessarily apply. So for those plots, the square plots are totally fine. And then they have some other points here. So this just finishing off those 10 points. The first one is finding the hidden signal, right? So PCA, as I mentioned, linear method, useful in reducing the noise by transforming the data. However, it's still trying to fit these like ellipsoids and trying to find these axes of these ellipses. So it's modeling the global structure. Useful for any data. I highly recommend applying it just to see what comes out of it in terms of the visualizations. Very interpretable visualizations. t-SNE more is a nonlinear method. It models the local structure, right? So the individual clusters that you find means that they're very similar, but there's no meaning to the distance between clusters. You commonly used for single cell data, and there's also UMAP as well. FATE is also another method that models both local and global structure. Very useful for this tree-like looking shape for like differentiation patterns in single cell data. Tip number 9 is on multimodal data, which we'll talk about on Monday. And then there's this last comment about keeping in mind like how robust really are these results, right? Because a lot of focus and a lot of interpretation has gone into like really trying to understand these clustering patterns for these approaches, and hopefully you got like sort of a sense of how variable they can be depending on the approach you're applying to it, right? So take it with a grain of salt and don't assume you found a rare cell type or something based on some scatter plot that you identified using like one method, for example. A comment here on how outliers can affect dimension reduction methods. And there are robust variants out there. So there's this thing called robust PCA. I linked this to an R package, which tries to associate, it tries to penalize observations that are far from like the center of this cloud. And so if you have like a presence of very strong outliers, then some of these robust methodologies are very useful. And this is just a summary of those 10 points around effective dimension reduction approaches. And I try to go into each of them in more detail. But I think the main takeaway would be looking at really the type of data that you have, the scale that's on, and really using the right methodologies for those appropriate data sets. And yeah, so with that, I'll come to a close, and I'll take any questions, or have if you have questions about the notebook, happy to answer that as well. I actually didn't go over the code just because I thought I would run out of time. So if anyone has any questions on the code itself, you can let me know.
There's a question from Jeremy.
Well, I guess it depends on your mixed data type that you have, right? So if it's I guess maybe you're talking about like SNP data, or if it's just clinical, then it really depends on how much mixing there is. Like if most of it's continuous, then turning some of the categoricals into continuous and applying PCA is going to be very similar to MFA. But if you have like an equal mix, I would say, then MFA is probably better to go with. But if you have all categorical, then you want to use MCA, which is multiple correspondence analysis. Okay, there's no questions. I guess I can just stick around. If people have questions, they can ask me.
Yeah, that works. And then in the afternoon, we'll be going over what's the
It's a
content?
biomarker
Biomarker.
discovery.
Okay, cool. Yeah, so we'll stop the recording here and people are welcome to stay and ask questions. If not, we will see you again at 12 o'clock.
I didn't go through the code, but I since I'm like under 30, like half an hour, I'm wondering if I should go through the code like for the next session at least.
Sure.